Your pillow has gone warm on one side. You shifted at some point, maybe minutes ago, maybe longer, and the cool side is already gone, already matching your temperature, and that small fact is barely a thought. It's just something the skin registers, the body making its quiet adjustments, while the mind does what it does at this hour, which is mostly drift. Most of the brain is drifting too. What happens in the sleeping brain is, by most measures, a kind of organized retreat. Not silence, the brain is never fully silent, but a drawing inward. A reduction of certain signals, a permission, chemical and electrical at once, for whole regions to slow their firing, to stop attending so urgently to the world outside the skull. The thalamus gates incoming sensation. The sensory cortices grow quieter. The planning regions, the ones that spend the day calculating and anticipating and second-guessing, they begin to step back from the threshold of awareness. Most of them, there's a region, roughly behind your forehead give or take, that doesn't always cooperate with this retreat. The prefrontal cortex is its clinical name, though the name doesn't quite capture what it does, which is something closer to watching, evaluating, holding the current moment up against a model of what the current moment is supposed to be. It is among the most metabolically expensive tissue in the human body. Evolution spent a very long time building it, and during ordinary sleep it powers down with everything else, taking its considerable energy demands somewhere quieter. Except sometimes it doesn't. Not loudly, not in a way you'd notice if you weren't looking for it. But in certain sleepers, on certain nights, something in the prefrontal cortex stays lit, partially, subtly, in a way the instruments can see even when the sleeper themselves can't yet. The EEG electrodes pick it up, a frequency that doesn't quite belong to the slow-wave landscape of deep sleep. A small, persistent flicker of something that looks electrically like waking. It's strange to think about. The brain, by all the usual indicators, is asleep. Heart rate slow, muscles slack, eyes sealed, and yet, and this is the part that researchers in sleep labs have now recorded in enough subjects that it is no longer considered anomalous. Something up there is still watching. The question of why this happens does not have a single clean answer, which is maybe appropriate because the brain itself rarely has single clean answers for anything. Part of what's understood is this. During REM sleep, the phase of sleep most associated with dreaming, with the vivid, strange, emotionally textured experiences that vanish so quickly on waking, the brain is not idle. It is in certain ways more active than it is during some waking hours. The visual cortex fires. The limbic structures, the amygdala especially, the brain's oldest threat assessor, run hot. The motor cortex sends signals that the brainstem carefully blocks before they reach the muscles. The body is held still while the brain generates experience from the inside. But the prefrontal cortex during ordinary REM tends to go quiet. The dreaming brain is, in this sense, a brain without a supervisor. Emotion runs, narrative runs, memory fragments surface and recombine without editorial constraint. You find yourself in a house that is somehow 3 houses at once, and it seems entirely normal, because nothing is checking it against the model of how houses work. This is standard, expected. The default condition of the dreaming brain. What is not standard is when the prefrontal cortex comes back. Not fully, never fully during sleep, but enough. Enough that the dreamer begins to notice something off about the experience. A door in the wrong place. A person who was somewhere else a moment ago and is now here. The light doing something light shouldn't do. And instead of absorbing the inconsistency into the dream's logic, as the unsupervised brain would normally do, something catches. Something that feels from the inside like a question forming. This isn't quite right. That thought, more tentative, arriving without fanfare, is what the biology is building toward. The moment before lucidity, the prefrontal cortex, still only partially online, beginning to run its usual function, checking the current moment against a model of what the current moment should be. The model says this is wrong. What follows from that depends on the sleeper, the night, the depth of sleep, the particular history of the brain doing the noticing. Some people wake up immediately. The checking triggers an arousal response, the dream dissolves, and they lie in the dark, reconstructing what they can before it fades. Others tip forward into something else entirely, something rarer. A state that has been documented, studied, measured, and still feels, to most people who experience it and even to some researchers who study it, like something the brain should not be capable of. Awareness, sustained, inside a dream. The instrument traces that record it are not ambiguous. You can see the prefrontal activity. You can see the gamma waves, fast, tight, associated in waking life with focused conscious attention, appearing in a brain that is simultaneously showing all the hallmarks of REM sleep. The two signatures together, gamma and REM, conscious attention and dreaming coexisting in the same skull, are what the science points to when it tries to say precisely what a lucid dream is. Not a half-waking, not a confused arousal. The body is genuinely asleep. The muscles are genuinely paralyzed by the same brainstem mechanisms that prevent you from acting out your dreams every night. The REM markers are present and clear, and inside all of that, the prefrontal cortex is running something, checking something, generating a kind of self-awareness that during ordinary dreaming the brain specifically switches off. I find it difficult, honestly, to absorb that without pausing, not because it's dramatic, it isn't. In the way of dramatic things, it's quiet. A few microvolt differences in the electrical trace. A region lit where it is usually dark. But what that lit region represents, what it means for the dreamer lying in the sleep lab bed, eyes moving rapidly behind closed lids, occasionally sending the agreed-upon signal, 2 deliberate left-right eye movements, a private Morse code from inside unconsciousness. What it means is that something in the brain has achieved a position that feels almost impossible by the rules of sleep. It knows it is dreaming, not guessing, knowing. In the same way that waking awareness knows the difference between a memory and the present moment, between imagining a sound and actually hearing one. The distinction is real, the brain is making it, and the brain is doing this while, by most available metrics, it is asleep. There is something the researchers have noted in subjects who lucid dream regularly, something that takes time to accumulate, months in some cases, before it becomes visible on the scans. The prefrontal cortex in experienced lucid dreamers shows signs of being, in a structural sense, slightly different from matched controls who dream without awareness. Not dramatically, not in ways that are obvious on a standard clinical image, but measurable. The tissue that stays lit on those particular nights may, over time, be changed by its own unusual activity, the brain, as it always has, adjusting to what is asked of it. What is asked of it here is strange by any biological standard. Stay asleep, keep dreaming, and simultaneously keep one part of yourself awake enough to know that none of this is real. The pillow beneath your cheek is real. The warmth that has gathered there since you stopped moving, that's real. The slow weight of your own breathing, the barely there sound of the room settling around you. The way the body has gone heavy in a way it only goes at this particular hour, that's all still here. The brain, somewhere behind your forehead, is beginning its own retreat. The supervisory regions stepping back from the threshold. The checks and comparisons running slower, then slower still. Most nights, the prefrontal cortex goes all the way down with everything else. Most nights. The equipment in a sleep lab at night makes a particular sound, not loud. A low, steady electrical hum. The polysomnograph running, the pens or the pixels tracing whatever the brain is doing, continuous and indifferent. The researchers who run these studies learn to stop hearing it after a while. It becomes the ambient condition of the work. On a night in 1975 in a lab in Hull, England, that hum was recording something it had never recorded before. A man named Alan Worsley was asleep. His REM markers were unambiguous. The EEG showing the fast, irregular waves, characteristic of dreaming sleep. The muscle atonia confirmed, the rapid eye movements visible on the trace. By every criterion available to the science at the time, he was dreaming. And dreams, at that point in the history of sleep research, were considered unreachable. You could watch someone dream from the outside. You could not receive a message from inside one. Worsley had agreed, before falling asleep, to move his eyes in a specific pattern the moment he became aware he was dreaming. Left, right, a signal. The trace recorded it. 2 deliberate lateral eye movements, distinct from the irregular scanning of ordinary REM, arriving in the middle of an otherwise uninterrupted dream state. Keith Hearn, his researcher, saw it happen in real time. It's worth sitting with what that moment actually was, scientifically. Not the romance of it. The romance comes easily and isn't the point. The point is that a boundary was crossed. The assumption had been that the dreaming mind and the waking world were, during sleep, categorically unreachable from each other. The dreamer was in there. The researcher was out here. No signal could pass between them, because the dreamer was unconscious, which meant there was no sender. Worsley was not unconscious. That's what the eye movement meant. That somewhere inside a brain that was, by all physiological standards, asleep, there was something oriented enough to remember an agreement made hours earlier, notice the right moment, and execute a deliberate motor act using the one muscle group the sleeping brain doesn't fully paralyze. The eyes, always the eyes. The brainstem during REM sends inhibitory signals down the spinal cord, a careful, precise suppression of voluntary muscle movement that prevents the sleeper from physically enacting whatever the dream contains. It's protective. Without it, the dreaming brain would move the body. The motor cortex fires during dreams in ways that closely mirror waking movement. The suppression is what keeps the body still, but the extraocular muscles, the ones that move the eyes, are exempt. They remain active during REM. Nobody fully agrees on why. Some researchers think the eye movements are related to the dreamed visual scene, the sleeping brain tracking the dream's imagery as it would track a real scene. Others are less certain. The mechanism stays a little murky. What matters is the gap it left open. Hearn's work went largely unnoticed at first, published quietly, poorly distributed, absorbed into the narrow literature of a field that wasn't yet sure what to do with the finding. A few years later, Stephen LaBerge at Stanford arrived at the same result independently. The same eye movement protocol, the same confirmation, the same overturning of the assumption that the dreaming mind was sealed off from contact. This time it was noticed. LaBerge spent the following decades building a research program around the finding, developing induction techniques, training subjects, accumulating the physiological data that would eventually give lucid dreaming its foothold in mainstream sleep science. The eye signal became a standard, a grammar, a way for the dreamer to say, "I'm here." 40-something years of that signal now, recorded in labs across several countries, in sleepers of varying ages and backgrounds and neurological profiles. The trace always looks the same, 2 deliberate lateral movements in a sea of irregular ones, arriving without warning from inside. What the researchers watching those traces in real time describe is not excitement usually. It's something quieter, a recognition. The machine confirming what the sleeping person already knew, that they were somewhere, and they knew it, and they reached back. The hum of the equipment continues. The trace keeps moving. The dreamer, on the other side of the signal, is already somewhere else. Your body is very heavy right now, not uncomfortably so, just settled. The kind of weight that arrives slowly without announcement somewhere around the 2nd or 3rd hour of the night, when the muscles have finally stopped holding themselves ready for anything, and have gone somewhere past relaxation into something more complete. You're not trying to stay still. The stillness is just happening. The body has made a decision you weren't consulted about, and the decision was enough. This is not nothing biologically. The stillness is produced. It costs something. There are neurons in the brainstem, a specific cluster, located in a region called the ventral medulla, that are, right now if you're drifting, actively firing inhibitory signals down through the spinal cord, landing on the motor neurons that connect to your skeletal muscles and suppressing them. holding them below the threshold they'd need to generate movement. The stillness isn't absence, it's the result of something working hard to create it. And above that, in the brain itself, nothing like stillness. A single neuron in the visual cortex during REM fires in patterns that would not look on a recording, obviously different from the patterns it makes when your eyes are open and you're watching something move. The action potentials arrive at roughly the same frequency, the same electrochemical cascade, sodium rushing in through membrane channels, the interior of the cell briefly reversing its charge, then potassium pushing out to restore it, the whole sequence taking about a millisecond, then resetting, then happening again. The machinery is identical. What's changed is the source of the signal. During waking, the signal driving that visual neuron comes ultimately from the outside. Light hits the retina. The retinal cells transduce it. The signal travels back through the optic nerve, through the lateral geniculate nucleus of the thalamus, and arrives at the primary visual cortex already shaped by what the eye actually saw. The neuron is, in a loose sense, reporting. During REM, the thalamic gate has narrowed. The signal from the retina isn't making it through cleanly, and yet the visual cortex is still firing, not randomly, not in noise, but in organized, coherent patterns, because the signal is coming from elsewhere, from higher cortical areas sending activation downward, from the limbic system pushing emotional content into the visual processing stream, from the brain generating imagery from the inside rather than receiving it from the outside. The neuron doesn't know the difference. It can't. A sodium ion doesn't carry a tag identifying where the instruction came from. The membrane depolarizes because it was told to depolarize, and the origin of the instruction, retinal input or internally generated cascade, doesn't change the chemistry of the response. Your visual cortex, during a vivid dream, is doing something very close to what it does when you are watching something real. The experience of seeing at the level of the cell is nearly the same. Nearly. What's different is harder to locate. It lives somewhere in the organization of the firing, in the patterns across populations of neurons, rather than in any single cell's behavior. The top-down signal that drives REM vision is not quite the same as the bottom-up signal that drives waking vision. The timing is slightly different, the spatial precision slightly lower, the signal arriving in a sequence that the brain's own predictive machinery generated, rather than one shaped by actual photons. Researchers can distinguish the two states on a recording if they know what to look for. The neurons themselves are not making that distinction. There's something about that I keep returning to, in the particular way you return to facts that feel like they should change something about how you understand yourself. The cell is doing its job. The job looks the same from inside the cell, regardless of whether the world behind the information is real or constructed. The feeling of seeing, whatever that feeling is, however it arises from electrochemical events, is not diminished by the absence of an actual scene. What the dreaming brain sees, it sees completely. The same argument holds for the motor cortex. During REM, the motor cortex fires. Studies recording directly from motor neurons during sleep in animals and inferred from imaging in humans show activation patterns that correspond to movement sequences. Running, reaching, the fingers doing something. The signals go out, travel toward the muscles, and meet the brainstem's suppression before they can complete the circuit. The body stays still. But the movement happened electrically. The neurons rehearsed it. Rehearsed is not quite a metaphor. The word implies intention, and there is no evidence of intention in a sleeping motor cortex, but the firing pattern looks, structurally, like a rehearsal, like the cortex running a sequence it knows without running it to completion. What this does to the motor system over time, whether repeated REM activation of movement sequences does anything lasting to the circuits involved, is something that becomes more interesting when you add the ingredient of awareness. The body is still very heavy. That hasn't changed. The brainstem suppression that's holding your muscles below threshold is running regardless of anything happening above it. The paralysis is not responsive to dream content. A dreamer in the grip of the most vivid, action-filled dream is held just as still as one drifting through something quiet. The suppression doesn't modulate for intensity, but the cortex above, the visual areas, the motor areas, the limbic structures adding emotional texture to everything, so miso, that's running at something close to full capacity. Doing what it does during wakefulness, with the volume at nearly the same level, in a body the brainstem has decided will not be moved. The cell in the visual cortex fires again. Sodium in, potassium out, a millisecond of reverse charge, then restoration. The signal travels to the next cell, then the next. The body below doesn't move at all, the brain above doesn't stop. Sleep is mostly slow. That's not a simplification, it's the electrical reality. The sleeping brain, measured from the scalp, produces waves, and the waves that define the deeper stages of sleep are long, unhurried, rolling through the cortex at somewhere between 0.5 Hz and 4 Hz. Delta waves. Their slowness is the point. They are the brain doing something it cannot do while awake, a kind of large-scale synchronization, whole populations of neurons rising and falling together, creating the conditions for certain kinds of cellular repair and memory consolidation that faster, more fragmented waking activity can't accommodate. The slowness is useful. The slowness is the work. You've probably had the experience of a sound arriving too clearly in the early part of sleep, a door, a voice, something outside, and the way it lands differently than it would mid-afternoon. Too sharp, too present. The brain isn't buffering it the way it does during full wakefulness. It hits closer. Then the slow waves reassert themselves and the sound recedes, and you're back under. And the sharpness was just a brief return to the surface. That sharpness has a frequency. Around 40 Hz, give or take. Oscillations happening 40 times per second in the electroencephalogram trace tighten fast compared to the long delta rolls beneath them. Gamma. It's associated in the waking brain with focused conscious attention, with the binding of sensory information into coherent perception, with the moment when the brain is not just processing but integrating, pulling together inputs from different cortical regions and producing something unified out of them. Gamma, in waking, means something is being attended to. In ordinary sleep, it mostly disappears, which makes sense. The conditions that produce it, active attention, cross-regional integration, the brain holding multiple streams of information together simultaneously, are not the conditions of sleep. The slow waves take over, the gamma recedes. This is expected. This is what the data showed consistently for decades of sleep EEG research. Then the lucid dreamers went into the lab. What the recordings showed in subjects who were verified lucid, confirmed by the eye movement signal, the REM markers unambiguous, was a sustained increase in gamma activity, specifically in the frontal and frontolateral regions of the cortex. Not a flash of it. Not the brief gamma burst that sometimes appears in ordinary REM and fades within seconds. Something more persistent. A gamma signature sitting on top of the REM state. The two coexisting in a way that the earlier model of sleep would have described as contradictory. Ursula Voss and her colleagues published this in 2009. The finding was specific enough to be uncomfortable to ignore. The gamma increase was localized, not spreading across the whole cortex, but concentrated in the prefrontal and frontolateral areas, the same regions associated in waking research with metacognition, with the brain's capacity to observe its own processes. 40 oscillations per second in a sleeping brain, in tissue that was, by all the slower metrics, deeply in REM. What produces gamma is not entirely settled science. The leading account involves interneurons, a class of inhibitory neurons whose rapid firing helps synchronize the activity of larger neural populations, creating the conditions for the fast oscillatory rhythm. These interneurons are particularly dense in the prefrontal cortex. They are also, as it happens, among the cell types most sensitive to certain anesthetic agents, which is part of why anesthesia suppresses gamma so reliably, and why the return of gamma during emergence from anesthesia has been studied as a potential marker of returning consciousness. The connection between gamma and consciousness is not clean. Scientists are careful about it, because the history of neuroscience is full of single metrics that seem to track consciousness until they didn't. Gamma is not a consciousness signal. It is a signal that appears when certain kinds of conscious attention are happening, and it is absent when they're not, and the relationship between the two is still being worked out. But in the sleep lab, at 40 Hz, in a brain that a few minutes earlier had crossed into RM and then something shifted, began showing frontal gamma that hadn't been there before. The implication is hard to sidestep. Something changed in that brain. Something activated. And the activation looks electrically like waking attention. The slow waves haven't stopped. The delta is still there, still rolling, still doing what deep sleep delta does. The gamma is sitting inside it, not replacing it, just present. Two rhythms running in the same tissue, one belonging to sleep, one belonging to something else. The brain that produced it is still, by most measures, asleep. Whatever the gamma is marking, it arrived without the brain waking up. It arrived without the body waking up either. The muscle suppression is still running, the eyes are still closed, the heart rate has not changed. The brainstem is still doing its careful, precise work of keeping everything still. And somewhere in the frontal cortex, 40 times a second, something is oscillating that has no ordinary business being there. You are breathing right now without trying. You probably weren't aware of it a moment ago. The breath was just happening. The diaphragm contracting, the lungs expanding, the whole quiet mechanical cycle running on its own, as it has been running since before you were born, as it will keep running through whatever sleep comes next. And then something, this sentence or the one before it, brought your attention to it, and now you can feel it. The chest rising, the brief pause at the top, the slow release. That noticing is not nothing. It required something of the brain, a shift of attentional resources toward an internal process that was previously running beneath the threshold of awareness. The brain turned briefly to look at itself. This is, in miniature, what metacognition is. Not the grand philosophical version, not consciousness contemplating consciousness, not the self asking what the self is made of. Just the ordinary functional version, the brain monitoring its own states, flagging discrepancies, holding the current moment up against a model of what the current moment should contain. You do it when you realize you've been reading the same sentence for the third time without taking it in. You do it when you notice you're anxious before you've identified what you're anxious about. The brain catching itself in the act of something. It's expensive, not in a casual sense, in a precise metabolic one. The prefrontal cortex, which does the heaviest lifting in metacognitive tasks, consumes glucose at a higher rate than most other cortical regions. It is among the last structures to fully mature, not reaching its adult configuration until the mid-20s in humans, which is late developmentally by almost any biological standard. It is disproportionately large in humans compared to other primates, even after accounting for overall brain size. Evolution spent a great deal of time and energy building a cortex capable of watching itself think. The question the sleep lab data forces is a strange one. Why would that capacity, so costly to maintain, ever switch on during sleep? The short answer is that it mostly doesn't. Ordinary dreaming is, in the metacognitive sense, unsupervised. The dream runs, the dream is believed, the dreamer is inside it without any part of the brain standing outside it, checking its coherence against known reality. The prefrontal cortex has stepped back. The monitoring has paused. This is why dream logic holds, why the house that is 3 houses feels like a single house, why the person who died is present without any sense of wrongness, why the rules of ordinary experience are suspended without the dreamer registering the suspension. The brain has stopped watching itself, and in most sleepers, on most nights, this is where it stays. But the brain is a plastic thing, in the precise biological meaning of that word. Not flexible in the casual sense, but genuinely alterable by experience, by repetition, by the particular demands placed on it over time. Neurons that fire together, as the shortened version of Hebb's rule goes, wire together. Synaptic connections are strengthened by use. Pathways that are repeatedly activated become gradually more efficient, lower threshold, faster conduction, more reliable in their firing. This is learning at the cellular level. This is how the brain changes in response to what it is asked to do. What experienced lucid dreamers appear to have done over many months or years of practice is ask their prefrontal cortex to do something it was not designed to do at night, and it adapted. The evidence comes from structural imaging, MRI studies comparing the brains of experienced lucid dreamers to matched controls who dream without awareness. The differences are not large. They are not the kinds of differences that would flag on a clinical scan or alarm a radiologist, but they are there, in the measurements, in the right places. The prefrontal cortex in experienced lucid dreamers shows, on average, greater grey matter volume in regions associated with self-monitoring. The frontopolar cortex, a subdivision tucked at the very front of the brain, associated with prospective memory and the ability to hold a self-directed goal in mind while doing something else, shows measurable differences in experienced lucid dreamers compared to those who have never achieved the state. Prospective memory, the ability to remember to do something in the future while currently occupied with something else, which is, if you think about it, almost exactly what the lucid dreamer's brain is doing. It fell asleep, it entered the dream, and somewhere in the middle of REM it remembered, without being prompted, without an external cue, that it had intended to notice when it was dreaming. The frontopolar cortex remembered the plan. This is not the same as saying lucid dreaming made these brains different. The causality is genuinely unclear. It may be that brains with these structural characteristics are more naturally prone to spontaneous lucidity, and that the same people who experience it spontaneously are also the ones who practice it deliberately. The imaging studies available so far cannot fully disentangle predisposition from development. The researchers are careful about this. I think being careful about it is right. But the correlation is there, the tissue is different, and the difference is in the regions you would predict if you were designing a brain that had practiced noticing itself in the dark. What is particularly strange, sitting with this, is that the training didn't happen at night. Not primarily. The techniques that experienced lucid dreamers use to increase their frequency of lucid dreams, reality checks performed during the day, prospective memory exercise, the deliberate cultivation of a habit of questioning whether the current moment is real, these are daytime practices. The brain is being asked, during waking hours, to repeatedly perform a specific kind of self-monitoring, to pause and check, to hold a question in mind, is this real? The practice is waking. The effect shows up in sleep, which means that whatever cellular changes accumulate from repeated metacognitive practice during the day are somehow general enough to influence what the brain does in a completely different state at night. The monitoring habit, trained into the waking cortex, crosses the threshold into sleep, follows the brain down into REM, persists partially in tissue that should, by all evolutionary logic, have stopped monitoring hours ago. The prefrontal cortex, shaped by what was asked of it during daylight, carries something of that asking into the dark. You are still breathing without trying, the diaphragm contracting, the lungs expanding, the whole cycle running beneath whatever attention you can still bring to this hour, patient and automatic, as it has always been. The brain monitoring it, lightly, the way it monitors everything, not urgently, not with effort, just maintaining a soft awareness that things are proceeding as expected. That soft awareness is, tonight, the last thing to dim. It will dim, the monitoring will slow, the prefrontal cortex will step back from whatever it's been attending to and join the general retreat into sleep. Most nights, for most people, it goes all the way. In some brains, on some nights, it doesn't quite make it. And the tissue that stays partially lit, the same tissue that spent years being asked to notice, to check, to hold a question, keeps doing what it learned. Your hands are probably still right now, wherever they've come to rest, against the mattress, folded somewhere near your chest, lying open at your sides, They've gone slack in the way hands go slack only when the body has genuinely stopped organising itself around a task. The tendons are loose. The small intrinsic muscles of the palm, the ones that spend the day making fine adjustments you never notice, have gone somewhere past tension into a kind of passive weight. The hands are just resting there, heavier than they seem during the day, and they are not being asked to do anything. The motor cortex above them is another matter. There is a strip of tissue running roughly ear to ear across the top of the brain, the primary motor cortex, M1 in the shorthand, that maintains a detailed, almost absurdly specific map of the body's movable parts. The map is not proportional to size. The hands occupy a disproportionate region of it, the fingers especially, because the hands are among the most finely controlled structures into the human body, and fine control requires cortical real estate. More neurons, more connections, more dedicated circuitry per square centimetre of hand than per square centimetre of, say, the back. When a pianist plays, this map is active. The signal runs down through the corticospinal tract, a long descending pathway, axons bundled together, travelling from cortex to spinal cord to motor neuron to muscle fibre, and the keys are pressed. But when a pianist only imagines playing, sits quietly, eyes closed, runs the piece mentally without touching anything, the motor cortex fires anyway. Not identically to actual playing, not with the same amplitude or precision, but in recognizable patterns, the same regions, the same rough sequence, the cortical map of the hand activating as if the hand were moving, while the hand sits still. This has been documented carefully enough that it is no longer considered surprising. Motor imagery, the deliberate mental rehearsal of movement, produces measurable cortical activation. Sports psychologists use it. Rehabilitation programs use it. Musicians use it. The finding is robust across many studies, many modalities, many kinds of movement. The brain, when asked to imagine doing something physical, partially does it. Partially. The signal goes out from the motor cortex, it travels toward the muscles, and somewhere in the descending pathway, through mechanisms that are still not entirely clear, it falls short of the threshold needed to produce actual movement. The muscles don't contract, the fingers don't move, but the cortical part of the loop runs. The neurons fire in sequence. The synapses between them experience something, not the full weight of actual movement, but something in the same direction. Synapses strengthen with use. This is not a metaphor. It is the cellular mechanism underlying learning, the AMPA receptors on the postsynaptic membrane becoming more numerous or more sensitive in response to repeated activation, the connection between two neurons becoming more reliable because it has been used repeatedly. This process doesn't require that the movement be completed. It requires that the circuit be activated. Mental rehearsal activates the circuit, incompletely but genuinely. Now consider what happens to that circuit during sleep. The relationship between motor learning and sleep has been studied long enough to have produced a fairly consistent picture. A skill practiced during the day, a finger tapping sequence, a new movement pattern, a practice coordination task, improves overnight. Not gradually during the day, but in a step that occurs specifically with sleep. The improvement is not proportional to rest. It requires sleep. More specifically, it is associated with the slow spindles of NREM sleep and the REM cycles that follow. The brain, across the night, doing something to the freshly used motor circuits that practice alone doesn't accomplish. The consolidation is the word researchers use. The synaptic changes begun during practice are consolidated during sleep, stabilized, integrated, made less vulnerable to interference. The motor cortex fires during REM, as the previous chapter described, in patterns that resemble the practice movement. The sequence is rehearsed again, electrically, without the body being able to act on it. And by morning something has shifted. The fingers move more cleanly. The timing is tighter. The skill is improved in the night while the hands were lying still. Mental rehearsal starts the circuit. Sleep finishes the work. which is where the lucid dream enters and where the biology becomes genuinely harder to make clean claims about, though the direction of the evidence is not difficult to follow. During a lucid dream, the dreamer can direct the dream's content, not always reliably, not with perfect control, but with enough influence that experienced lucid dreamers report being able to practice physical skills within the dream. A golf swing, a piano passage, a dance sequence, a climbing movement, with a sensory vividness that mental imagery during wakefulness does not quite reach. The dream provides a simulated body, a simulated environment, a simulated resistance and feedback. The dreamer swings and feels the swing, the dreamer plays and hears something like sound. The simulation is generated by the same brain that would process the real thing, using the same sensory cortices, the same motor planning regions, the same proprioceptive processing. Whether the motor cortex during lucid dream practice fires in patterns closer to actual movement than mental imagery produces, that question has not been answered cleanly. The technical difficulty of obtaining precise motor cortical recordings during sleep in subjects who are simultaneously maintaining enough prefrontal awareness to direct their dream content is substantial. What exists is EEG evidence of elevated frontal gamma during lucid dreaming, the general REM motor cortex activation finding, the mental imagery literature showing partial motor activation, and case reports from experienced lucid dreamers describing improvement in waking skills following dream practice. None of this is proof of mechanism. It is a coherent set of pieces pointing in the same direction. The direction is the motor circuit is being activated during the lucid dream. The dream provides a richer, more embodied simulation than waking imagery. The REM state during which the practice occurs is precisely the state associated with motor memory consolidation. And the synaptic machinery that responds to Aktivituden, that strengthens connections when circuits are used, does not distinguish between sources of activation in the way we might intuitively expect. The neuron fires because it was told to fire. The synapse strengthens because it was activated. Whether the activation came from a real piano or a dreamed one is not information the synapse receives. Your hands are still lying wherever they came to rest. The motor cortex above them is quiet or close to it. The waking signals have thinned, the day's movement patterns no longer running through the circuit. Soon, if sleep comes, the NREM spindles will begin their slow work on whatever the hands practice today. Small things, maybe. Typing, a door handle turned a particular way. The specific grip of pulling a blanket up at some point in the evening. Small circuits, but circuits that were used. Circuits the sleeping brain will return to. And if the brain tonight finds its way into a lucid dream, if the prefrontal cortex stays partially lit, if the gamma appears, if something in there becomes aware, then whatever the hands do in that dream, whatever the motor cortex rehearses in the warm, immersive simulation of a dreamed room, the dreamed weight of dreamed objects in dreamed palms, the synapses will not know the difference. They will only know they were used. The amygdala is old. Not old in the way that feels like a useful fact, old in a way that changes the meaning of everything around it. The limbic structures at the brain's core, the amygdala among them, were present in vertebrate nervous systems long before the cortex had done anything resembling what it does now. Long before language, long before planning, long before any structure existed in the skull capable of reflecting on what the skull contained. The amygdala was already there, already doing this. What it does is not complicated to describe. It monitors, continuously below awareness, without being asked. Incoming sensory information passes through or near it on the way to cortical processing, and the amygdala is checking it, running a fast crude threat assessment that operates in milliseconds, well ahead of the slower conscious appraisal that follows. A shape at the edge of vision, a sound with an irregular pattern, a smell that belongs to something wrong. The amygdala responds before the cortex has finished deciding what the thing is. This speed is the point. The cortex thinks, the amygdala reacts. The distinction is not a failure of the amygdala, it is its entire biological purpose. In an environment where threats were physical and immediate, where the interval between detecting danger and responding to it determined survival, the amygdala's blunt speed was more valuable than the prefrontal cortex's careful deliberation. The deliberation could come after. First, the body needed to move. The chest knows this before anything else. Fear announces itself there, the sternum tightening, the breath changing, the heartbeat shifting its character, before the mind has assembled the reason for it. The amygdala has already sent its signal. The body is already responding to something the cortex is still processing. During ordinary dreams, the amygdala runs without the prefrontal cortex present to moderate it. This is part of why nightmare fear achieves intensities that waking fear rarely reaches. The amygdala generates the threat signal. The simulated environment responds to it, producing more threat, more signal. The loop runs forward without interruption because the structure that would normally apply context, that would weigh the threat against what is actually likely, that would ask whether the fear is proportionate, has stepped back. The prefrontal cortex, during ordinary REM, is largely offline. The fear has no one to report to. In the lucid dream, something changes. Not immediately, not reliably, not in every case, but the data from experienced lucid dreamers shows a consistent pattern. The amygdala activity during lucid REM is measurably different from its activity during non-lucid REM, not absent, not suppressed entirely, but modulated. The prefrontal cortex, partially reactivated, is doing something with the signal the amygdala sends. Not suppressing it exactly, more like receiving it, acknowledging it. The dreamer becomes aware of the fear while knowing they are dreaming. The chest still tightens. The signal is still real in the physiological sense. The amygdala is not fooled by the knowledge that the dream isn't real, because the amygdala doesn't process knowledge of that kind. It responds to the simulated threat as it would respond to an actual one. But above it, something is watching. The oldest structure in the brain, and among the newest, occupying the same moment in the same skull. The amygdala running its ancient machinery. The prefrontal cortex observing it, calm enough to observe, not calm enough to stop it entirely. Whether that observation changes anything lasting in the circuit between them, whether repeated lucid encounters with fear, watched rather than consumed by, alter the amygdala's threshold in some durable way, is a question the research is still building toward. The sternum is still. Whatever the chest knows at this hour, it is keeping quiet. The darkness behind closed eyelids is not quite black. It has a colour that doesn't have a name, something warmer than black, something that shifts when you press gently against the lid and changes again when you stop. It's the colour of the inside of something biological. Capillaries near the surface, the retina still faintly responsive to pressure. The whole apparatus of vision doing something low and quiet in the absence of light. Not nothing, just the baseline murmur of tissue that hasn't fully switched off. The brain behind it is doing something similar. Not resting, not waiting. Running a set of processes that waking hours don't have the metabolic space for. Slower, more thorough. More concerned with structure than with function. The day was about using the brain. The night increasingly looks like it's about maintaining it. Neuroplasticity is the brain's capacity to change its own physical structure in response to experience. The word gets used loosely, sometimes as though it means the brain is infinitely malleable, always improving, always capable of rewriting itself given the right input. The reality is more constrained and more interesting. Specific synapses strengthen when the neurons they connect are activated together repeatedly. Specific connections weaken and are eventually pruned when they go unused. The proteins at the synapse are in constant flux. AMPA receptors inserted into the postsynaptic membrane when a connection strengthens, removed when it weakens, the molecular composition of each synaptic junction quietly shifting in response to its history of activation. This happens during waking. Every skill practiced, every memory formed, every pattern of thought repeated enough times to feel habitual. These are synaptic changes. The brain you will have tomorrow is not identical to the brain you have tonight. The differences are small mostly, and located in particular circuits, but they are structural, physical, real, in the way that a worn path through grass is real, made by use, visible in the tissue. What sleep does to this process is not simply pause it. The evidence for sleep's active role in synaptic plasticity has accumulated across several decades and across multiple levels of biological analysis, from molecular events at individual synapses to whole brain imaging showing overnight changes in connectivity. One of the better supported accounts, the synaptic homeostasis hypothesis proposed by Tononi and Torelli, suggests that waking experience tends to strengthen synapses broadly, the brain engaged with the world, potentiating connections across many circuits simultaneously, and that sleep performs a kind of selective scaling down, not erasing but normalizing, returning the overall synaptic weight to a sustainable baseline while preserving the relative differences, the connections most strongly potentiated during the day staying stronger than the rest, the signal-to-noise ratio of the whole system improving because the noise has been quietly reduced. The pruning is not random. It is, as far as the research can tell, targeted. Synapses that were weakly activated or not activated at all during the preceding waking period are candidates for downscaling. The molecular signals that mark a synapse for pruning, complement proteins, microglial activity at the synaptic junction, are more active during sleep. The glial cells that carry out much of the physical work of synaptic remodeling are more active during sleep. The brain is not merely consolidating what the day built, it is also deciding through mechanisms that operate below any level of awareness what to keep. BDNF, brain-derived neurotrophic factor, a protein involved in synaptic strengthening and neuronal survival, shows fluctuating levels across the sleep cycle. Its release is influenced by neural activity patterns, and the particular activity patterns of REM sleep appear to promote its expression in certain regions. The hippocampus, heavily involved in memory formation, shows BDNF-related changes across the sleep cycle that correspond to the consolidation of recently formed memories. The molecular environment of the sleeping brain is not a neutral holding state. It is chemically active in specific ways at specific times in service of specific structural outcomes. None of this was designed with dreaming in mind. The plasticity machinery runs whether the night is dreamless or vivid, whether the REM cycles are rich with imagery or thin and quickly forgotten. The synapses are being worked on regardless, but experience shapes what those synapses are, and the dreaming brain is having experience. This is the part that is easy to pass over without feeling its full weight. The circuits activated during a dream are activated genuinely, not metaphorically, not approximately, but in the same electrochemical sense as circuits activated during waking. The visual cortex processing a dreamed image is doing what it does when processing a real one. The motor cortex rehearsing a dreamed movement is running its sequences through the same synaptic machinery it uses during actual practice. The emotional circuits responding to a dreamed threat or a dreamed grief or a dreamed relief are releasing the same neurotransmitters, activating the same downstream pathways, producing the same cellular conditions that waking emotion produces. And the plasticity machinery running through the night is working on the synapses that those activations involve. I find myself uncertain about what to make of this in an honest sense. The implication that dreamed experience leaves structural traces in the brain, that what is encountered in a dream participates in the same biological process that consolidates waking memory and waking skill, sits at the edge of what the current evidence can cleanly support. The direction of it is consistent. The mechanism is plausible in a specific way, not a hand-waving way. But the direct demonstration that specific dreamed events produce specific synaptic changes detectable the following day, that study has not been done cleanly yet, or not done in humans in ways that control for everything that needs controlling. What can be said is this: the brain during dreaming is electrically active in ways that would, during waking, produce synaptic change. The same molecular environment that processes waking experience is present during sleep. The mechanisms that consolidate motor learning and emotional memory are running during the same sleep stages in which dreaming occurs. The pieces are all there. Whether they add up to what they seem to add up to is a question the biology is still sitting with. The darkness behind your eyelids has probably stopped being something you're tracking. At some point in the last few minutes, the colour of it stopped being a texture and started being just the absence of waking, the warmth of it, the particular quality of your own closed eyes at this hour, familiar and unremarkable, the way all the body's surfaces become unremarkable when attention finally stops cataloguing them. The synapses at the hippocampal junctions are doing something, quietly, with no awareness of being observed, no response to whether the person they belong to is still attending to them or has drifted somewhere past attending. They were activated today. The machinery has noted it. The night is doing what the night does. The structure of the brain you will wake up with is being decided right now in the dark, in the tissue without you. The eyelids are thin enough that if you have ever watched someone sleep close beside you, you may have seen it. The faint, irregular tremor beneath the lid, the eyeball tracking something that isn't there. The small, visible evidence of a brain doing something private and unreachable. The movement is quick, unpredictable. Nothing like the slow drift of light sleep or the stillness of deep NREM. It jumps, changes direction, follows something. What it follows is still not entirely settled. One account holds that the rapid eye movements of REM track the dreamed visual scene, the sleeping brain scanning a fabricated environment the way waking eyes scan a real one. Studies recording eye movements alongside dream reports find partial correlations. Dreamers who report looking left show leftward movement on the trace. Dreamers describing a tennis match and show the lateral oscillation of someone watching a volley. The correspondence is imperfect, but it is there often enough to suggest the movements are not noise. The brainstem during REM sends its inhibitory signals down the spinal cord and silences the skeletal muscles. The diaphragm keeps working, the heart keeps working, and the extraocular muscles, the 6 small muscles anchoring each eye in its socket, responsible for every deliberate gaze shift during waking, keep working. Nobody fully agreed on why they were exempted, but they were, and that exemption left a gap. Keith Hearn saw it first, a gap between the dreamer and the lab. The dreamer sealed inside the dream, no way out, no way to signal across the boundary of sleep, except that the eyes were still moving, still responsive, still capable of deliberate direction if the dreamer was aware enough to direct them. Left, right, agreed upon before sleep, executed from inside. The polysomnograph recorded it as 2 sharp lateral deflections in a sea of irregular movement, distinct, unmistakable, a signal from somewhere the science had assumed was unreachable. What this required of the sleeping brain is worth pausing over. The dreamer had to maintain the memory of an intention formed before sleep, had to recognize the moment of lucidity when it arrived, had to locate within the dream the voluntary control of a muscle group that the brain was not suppressing, had to execute a specific pattern rather than a reflexive one. All of this in a brain that was simultaneously asleep. The eye moved because something directed it, something that knew where it was, what it had agreed to, and how to reach back. The lid is still. Whatever is happening behind it right now, the slow drift of early sleep or something faster, something that tracks. There is no way to know from outside. The eye keeps its own record. You know the feeling of catching your mind somewhere it wandered without permission. You were reading or listening or doing something that required the front of your attention, and then at some point, without deciding to, you were somewhere else entirely. Planning something that isn't happening yet. Replaying something that already did. Running a conversation that will probably never occur in quite the form you're rehearsing it. And then something pulled you back. A word landing differently, a sound from outside, the faint lurch of returning attention, and you were here again, slightly surprised by the gap. That wandering has a biology. When the brain is not engaged with a specific external task, not solving, not tracking, not responding to something demanding attention from outside. A particular network becomes more active, not less. This counterintuitive fact took researchers some time to accept because the assumption had been that the resting brain was simply idling, its activity falling towards some neutral baseline when nothing was required of it. What the imaging showed instead was that certain regions became specifically, reliably, more active during rest. Not random activation, a coherent network, switching on together, doing something. The network includes the medial prefrontal cortex, the part of the prefrontal cortex oriented inward, toward the self rather than outward toward the world. The posterior cingulate cortex, tucked near the brain's midline, involved in self-referential processing and the integration of autobiographical memory. The angular gyrus, sitting at the junction of several cortical regions, involved in pulling meaning from context. The hippocampal formation, threading memory into the present moment. These regions activate together reliably when the brain is given nothing specific to do. They are collectively the network of the wandering mind. The self-referential label researchers attach to the DMN, default mode network, named for the state in which it dominates, is accurate in a fairly precise sense. What these regions do together is something like maintain the ongoing narrative of the self, integrate past experience with present context, project forward into imagined futures, hold the continuous thread of personal identity that allows the brain to experience a coherent sense of being someone who has a history and an anticipated continuation. When you are doing nothing, this is what your brain is doing instead. During ordinary dreaming, the default mode network runs. The dream is, in many respects, exactly the kind of internally generated narrative the DMN specializes in. Self-referential, temporally loose, pulling from memory and projection simultaneously, building a world from the inside rather than receiving one from the outside. The DMN is well suited to dreaming. It does in sleep what it does in waking daydreams, only without the occasional interruptions that external reality provides. The dream in this framing is the default mode network with nothing pushing back against it, which makes the lucid dream something specific, a state in which something does push back. Imaging studies comparing lucid and non-lucid REM, technically difficult studies requiring subjects capable of reliably achieving lucidity in the scanner, but accomplished in enough subjects now to provide consistent data, show a pattern that fits this framing. During lucid REM, the prefrontal regions associated with self-monitoring and metacognition show increased activation relative to non-lucid REM, and the relationship between the prefrontal cortex and the default mode network shifts. The DMN doesn't disappear, the dream continues, the narrative continues, the self-referential processing continues, but something is modulating it. Something is sitting alongside the network's unchecked running and introducing a different signal. The interruption is not dramatic on the scan. It's a change in connectivity, in the balance of activation between regions, in the degree to which the prefrontal cortex is influencing rather than simply participating in the network's activity. But the behavioral correlate of that change, the subjective experience of suddenly knowing you are dreaming, of the narrative continuing while something observes it, is, by the accounts of everyone who has experienced it, anything but subtle. The dream keeps running. The DMN keeps generating its self-referential content. The narrative doesn't stop. But something has stepped outside it. This is what I find most biologically strange about lucidity, if I'm honest about where the strangeness lands for me. The default mode network is, among other things, the network of the self. The sense of being a continuous person with a history and a future is something this network actively constructs, not reflects, but constructs, from the materials of memory and projection and self-referential processing. During ordinary dreaming, the self the DMN constructs is inside the dream entirely, seamlessly, without any remainder. In the lucid state, the construction continues, but something has noticed that it is a construction. Not from outside the brain, there is no outside. From within the same skull, using tissue that is itself part of the same network it is now observing, the brain has generated a perspective from which the dream looks, briefly, like what it is, the lurch of returning attention, the small disorientation of catching the mind somewhere it wandered. You know that feeling already. What the lucid dreamer experiences is something in the same family, the same basic biology of self-monitoring suddenly active, except the wandering the brain catches itself in is not a daydream or a distraction. It is the entire constructed world of the dream. The default mode network keeps running. The dream doesn't end. The narrative continues forward, generating the next moment from the materials of everything the sleeping brain contains. But the interruption has occurred, and the brain for a moment is in two places at once, inside the story it is telling and just barely outside it, watching itself tell it, the way attention returns from wandering and finds briefly the gap between where it was and where it is. The mind wanders. Sometimes in sleep something notices. The throat tightens first, before the chest, before the stomach, before whatever the mind is doing with the thing that caused it. The throat, that narrow, unremarkable corridor, goes taut in a way that has nothing to do with swallowing and everything to do with the body bracing for something. You felt it. The specific quality of it, distinct from other kinds of tension, arriving with a speed that thinking doesn't match. Fear is a body event before it is anything else. The amygdala's signal reaches the brainstem and the hypothalamus in milliseconds, faster than the cortex has finished processing what triggered it. The hypothalamic pituitary adrenal axis begins its cascade. Corticotropin-releasing hormone travels a short distance and prompts ACTH release into the bloodstream, which reaches the adrenal glands sitting above the kidneys, which release cortisol. The heart rate climbs, the bronchioles dilate. Blood shifts toward the large muscles of the limbs. The body is preparing for something physical before the mind has decided whether the threat is real. This sequence doesn't ask for permission. During a nightmare, it runs fully. The dreamer is asleep, the body paralyzed, the muscles suppressed by brainstem inhibition, the eyes moving behind sealed lids, and the cortisols rising anyway. The heart rate is genuinely elevated. The amygdala is treating the dreamed threat as it would treat a waking one, because the amygdala does not receive information about the source of the signal it's responding to. The visual cortex fires, the emotional circuits fire. The hypothalamus receives what looks to it like a real threat report. The body responds to the dream as if the dream were happening. Now the dreamer becomes aware they are dreaming. The cognitive shift is real and sudden, the prefrontal cortex partially reactivating, the metacognitive awareness arriving, the knowledge crystallizing that none of this is real. The dreamer knows. This is the moment that feels, from the inside, like it should change everything. The threat is fictional. The dreamer is aware of its fictionality. Surely the body should stand down. Often it doesn't. Not immediately, not completely. The throat is still taut. The heart rate is still elevated. The cortisol already released into the bloodstream is already there, already binding to its receptors, already doing what cortisol does. Because cortisol, once released, doesn't unrelease because someone upstairs has updated their threat assessment. The HPA axis cascade has a momentum that cognitive knowledge cannot simply reverse. The body is still in the fear. The mind knows it's a dream. These two facts coexist uncomfortably in the same skull. This gap between knowing and feeling is not a failure of the lucid dreamer. It is information about where emotion lives. Emotion is not a cortical event that the body enacts. It is a body event that the cortex sometimes catches up to. The amygdala's response to threat is largely independent of cortical appraisal. It runs through pathways that don't route through the prefrontal cortex on their way to producing a physiological response. The cortex can modulate the amygdala. Over time, with repeated experience, with the particular kind of exposure that therapists use in treating anxiety disorders, repeated contact with the feared thing in a context where the catastrophe doesn't occur, The cortical influence on amygdala response can increase. The threshold shifts, the response dampens, but in the moment, in the dream, the cortisol is already there. What experienced lucid dreamers report over many nights and many such moments is a gradual change, not in whether the fear arrives, but in its duration, its grip, the speed with which the body catches up to what the mind already knows. The amygdala still fires, the cascade still begins, but the interval between the fear's arrival and its recession appears to shorten in some accounts with practice. Whether this reflects actual change in the amygdala's threshold, or in the strength of prefrontal modulation pathways, or simply in the dreamer's learned behavioral response to the fear once it arrives. The biology has not cleanly separated these yet. The accounts are consistent. The mechanism remains somewhat open. What the data does suggest is that the cortex and the amygdala in the lucid dream state are in a different relationship than they are during ordinary dreaming. Not the cortex winning, not the amygdala silenced. Something more like a conversation, the amygdala sending its ancient signal upward, and something above it present enough to receive it, to hold it, to let it run its physiological course without the feedback loop that turns ordinary nightmare fear into an escalating closed system. The throat, in time, loosens, not because the dreamer decided it should, because the cascade completed, because the cortisol was metabolized, because the body, given enough time without additional threat signal, found its way back to baseline on its own. The knowing didn't stop the fear, it may have changed what the fear did next. Certain traditions treated the dreamer's preparation, not metaphorically, as genuine rehearsal for dissolution, for the loosening of the self from the body, for whatever the practitioners believe came after. Tibetan Buddhist dream yoga, practiced across centuries, used the lucid dream state deliberately. The practitioner trained to recognize the dream, and then, within it, to practice the release of the self's usual boundaries, to experience the dissolution of ordinary waking identity and remain present through it. The neuroscience does not confirm what the practitioners believed they were preparing for, but it confirms something adjacent, something cellular and measurable, which is a different kind of confirmation, narrower, quieter, making no claims about what lies beyond the biological. What it confirms is this: the brain can be changed by surviving something terrifying inside a dream. The mechanism belongs to a well-documented learning process called extinction. When a feared stimulus is encountered repeatedly in a context where the anticipated catastrophe does not occur, the fear response gradually dampens. Not erases, the original fear memory remains encoded, retrievable, capable of returning under certain conditions, but the response weakens. The amygdala's threshold for firing shifts. The prefrontal cortex's inhibitory influence over the amygdala strengthens along the pathway connecting them, a structural change produced by the repeated experience of threat perceived, threat survived, catastrophe absent. This is the biology behind exposure therapy, behind the gradual approach toward feared things that trauma treatment has used in various forms for decades. The brain learns, synapse by synapse, that the feared thing can be endured. The question the lucid dream raises is whether the feared thing needs to be real. The evidence from mental imagery research suggests it doesn't need to be fully real. Imaginal exposure, deliberately imagining feared scenarios in a therapeutic context, produces extinction learning. Weaker typically than in vivo exposure, but present. The amygdala responds to simulated threat, and the extinction process works on simulated threat because the amygdala is not receiving information about whether the threat is simulated. The lucid dream offers a simulation richer than deliberate imagery. The visual cortex is generating it fully. The emotional circuits are engaged. The body is producing genuine physiological responses. The cortisol, the elevated heart rate, the cascade that doesn't wait for confirmation that the threat is real. The dreamer falls, or faces what they cannot face, or moves through the moment of imagined dissolution, and then does not die, does not cease, wakes or continues dreaming, or simply survives the peak of the fear and finds the body settling back toward baseline on the other side of it. The amygdala notes the outcome, not consciously, not as a memory in any narrative sense, but the synaptic machinery that governs threat response is experience dependent, and this was an experience. The cortisol peaked and metabolized. The heart rate climbed and fell, the threat was encountered and the organism continued. Repeated enough times, this leaves a trace. The Tibetan practitioners were not working with fMRI data or synaptic models. What they observed across generations of practice was behavioral. Meditators who trained in dream yoga reported changes in how they met fear in waking life as much as in sleep. A loosening of the grip that the anticipation of dissolution exercised over ordinary experience. The neuroscience would describe this as prefrontal amygdala pathway strengthening. Extinction learning accumulated across many sessions, making the transition from feared state to survive state more efficient, the recovery faster, the baseline easier to find. Different descriptions, possibly the same change in the tissue. Your breath is doing something right now that it will keep doing without you. The moment is very close. Where the breath stops being something you're aware of and becomes simply something occurring. The body taking over entirely, the self that has been tracking all of this, following the thread of it through the night, beginning to lose its grip on the thread. Not dying, just the ordinary loosening that sleep requires. The tissue will remember, in its own way, what the night contained. The scalp is heavier than it seems during the day. Or maybe it's just that at this hour, with the head resting against whatever it's resting against, the weight of it becomes something you can feel in a way that upright wakefulness doesn't permit. The skull, the tissue inside it, the slow electrical murmur of everything running beneath the bone, it all has mass. It presses down. And somewhere in that mass, right now, processes are underway that have been underway every night of your life, quietly adjusting the structure of the thing doing the adjusting. 60 days is not a long time in most biological contexts. A broken bone takes longer to heal fully. The complete turnover of certain cell populations in the gut takes longer. The hair has barely grown. And yet 60 days of a specific, repeated cognitive practice, a deliberate daily effort to alter the state of awareness during sleep, appears, in some studies, to be enough time for measurable structural differences to begin accumulating in the brain, not large differences. This is where the literature requires careful reading, because the popular accounts of neuroplasticity tend toward the dramatic, and the actual data tends toward the precise and modest. The changes documented in studies examining experienced lucid dreamers, and in the smaller number of longitudinal studies that have followed subjects through induction training programs, are measurable in the way that requires good imaging equipment and careful statistical analysis to find. They are not the kind of differences visible to the naked eye on a clinical scan, but they are in the right places. The frontopolar cortex, the anterior most region of the prefrontal cortex, sitting at the very front of the brain, shows increased grey matter density in experienced lucid dreamers compared to matched controls in several imaging studies. This is the region associated with prospective memory, with the ability to hold a self-directed intention in mind while engaged in something else, with the particular kind of metacognitive monitoring that asks not just what is happening, but whether what is happening matches what was intended. It is, in a structural sense, one of the regions you would most expect to find changed if the brain had been repeatedly asked to maintain an intention across the boundary of sleep. The white matter tells a different part of the story. White matter, the myelinated axon bundles that connect different cortical regions, allowing them to communicate with speed and reliability, shows changes in fractional anisotropy in experienced lucid dreamers along specific tracts. Fractional anisotropy is a measure of the directionality of water diffusion through tissue. Higher values generally indicate more organized, more coherent white matter structure, suggesting more efficient signal transmission along that tract. The tracks showing changes in lucid dreamers connect prefrontal regions to the regions involved in self-referential processing and memory. the same network, roughly, that the default mode network research implicates in the difference between lucid and non-lucid dreaming. These structural connections between these regions appear in experienced lucid dreamers to be slightly more developed. I want to be honest about what this does and doesn't establish, because the distinction matters. These studies are almost entirely correlational. They compare people who are already experienced lucid dreamers to people who are not, which means the structural differences observed could reflect the effects of years of practice, or they could reflect the predispositions that made those individuals more likely to experience and pursue lucid dreaming in the first place. A brain with a naturally more developed frontopolar cortex might be more prone to spontaneous lucidity, and the same person might be more motivated to practice deliberately. The structural difference and the lucid dreaming history would then correlate not because one caused the other, but because both followed from an underlying characteristic. The longitudinal studies, the ones that follow subjects before and after a training period, which is the design that can actually speak to causation, are fewer, smaller, and methodologically harder to execute cleanly. What they suggest, tentatively, is that structural change does begin to accumulate with sustained practice, but tentatively is the right word. The sample sizes are not large, the training periods vary, the outcome measures are not yet standardized across labs. What the 60-day framing captures is something the biology of learning more broadly supports, that the brain requires sustained repeated activation of a specific circuit before structural change becomes detectable. Single sessions leave functional traces, the synaptic changes that consolidation works on overnight. But the kind of change visible in the grey matter density measurements, the kind of change that shows up in fractional anisotropy along white matter tracks, requires more than a few nights of practice. It requires the brain to be asked the same thing repeatedly, across enough time that the asking becomes a shaping force. The brain responds to what is asked of it, not immediately, not dramatically, but with a biological fidelity that is, in its quiet way, remarkable. The tissue reorganizing itself around the demands placed on it, the connections between regions that communicate frequently becoming more efficient, the regions doing the most work becoming in measurable ways more developed. What was asked here was unusual by any evolutionary standard. The brain was asked to maintain waking-level metacognitive awareness inside a sleep state, to notice from within a fabricated experience that the experience was fabricated, to hold an intention formed before sleep across the threshold of unconsciousness and execute it inside a dream. The frontopolar cortex thickened slightly. The white matter connecting it to the self-monitoring network became fractionally more organized. The brain, asked to do something it was not designed to do at night, did what brains do with repetition. It became slightly more capable of doing it. The scalp is still heavy against whatever surface it has found tonight. The skull beneath it, the tissue beneath that. The slow electrical conversations running between regions that have spent the day being asked various things and will spend the night responding to those askings in the dark. All of it is still here, still running, still in the middle of a process that doesn't pause for sleep but deepens into it. 60 days from now, the brain will not be identical to the one pressing down into the pillow right now. It never is. What determines the direction of the change is what gets asked of it between now and then. Tonight, nothing is being asked. Tonight, it is simply running. There is a moment before waking that has no name. The alarm hasn't sounded yet. The dream is already thinning, not gone but losing its density, its confident insistence that it is the only thing. The body is beginning to return to itself without having decided to, the weight of the limbs becoming something feelable again, the specific pressure of wherever the head has been all night, the warmth gathered in the sheets, the particular quality of the room's darkness. These are beginning to re-register slowly, as if the senses are cycling back up from somewhere they'd been left running on low. The dream is still there, barely, and then it isn't. This is one of the stranger biological facts about dreaming, the speed with which the content vanishes. Not gradually, not like a memory fading over days, but within minutes of waking. sometimes within seconds, the narrative that felt completely coherent moments ago has collapsed into fragments, then into the vague impression of a tone, then into nothing retrievable at all. You know you were somewhere. You cannot find the somewhere. The reason belongs to neurochemistry rather than mystery. During REM sleep, norepinephrine, a neurotransmitter critically involved in the consolidation of explicit memories in the process of transferring experience from short-term to long-term storage, is largely absent from the cortex. The locus coeruleus, the brain stem structure that produces most of the brain's norepinephrine, goes quiet during REM in a way it does at almost no other time. The dream unfolds in a brain chemically unsuited to remembering it. The hippocampus, which would ordinarily be encoding the experience into retrievable memory, is running. It is active during REM. But without the norepinephrine signal that helps stabilize new memory traces, the encoding is shallow, partial, easily displaced by the neurochemical shift that waking brings. The dream dissolves because the brain was not, during the dream, building the kind of memory that survives waking. But the synapses were still changing. The cellular machinery of plasticity does not require norepinephrine in the same way that explicit memory encoding does. The AMPA receptor trafficking that strengthens synaptic connections, the BDNF release that supports synaptic consolidation, the slow synaptic scaling that sleep performs on the circuits the day activated. These processes were running through the night, working on whatever circuits the dream engaged, regardless of whether the experience would be remembered in the morning. The motor cortex that rehearsed a movement in the dream will not remember the dream. The synapse it used will be slightly different. This gap between what is remembered and what changed is the most precise answer the biology currently offers to the question this video began with. Can a lucid dream actually change your brain? The change is not stored where memories are stored. It is not accessible to the introspective mind that wakes up and tries to account for the night. The prefrontal cortex will stand down from whatever gamma flickering awareness it maintained during the lucid state. The dream will dissolve with the norepinephrine's return. The narrative will be gone within minutes, or will survive only in pale fragments that feel less like memories than like the residue of a mood. And in the tissue, quietly, something will be different. How different depends on many things: the depth and duration of the REM cycle, the intensity of the cortical activation during the dream, the particular circuits that were engaged, the accumulated history of previous nights doing similar things to similar circuits. A single lucid dream does not restructure the brain. The evidence doesn't support that. What it does is participate in the same incremental process that every night participates in, the slow, cumulative, largely invisible work of a brain adjusting its own architecture in response to what it experienced. The experience happened to be a dream. The adjustment doesn't know that. There is something worth sitting with in that, at this particular hour, when the night has been spent following the biology of a state most people will never intentionally enter, and some people cannot imagine achieving. The brain doing its cellular work in the dark, indifferent to the epistemological status of the experience driving the work, whether the pianist played or dreamed of playing, whether the feared thing was real or fabricated, whether the self that noticed the dream was dreaming, had any idea what its noticing was doing to the tissue that produced it. The tissue was doing what tissue does, using what arrived, adjusting to it, remembering it in the only language tissue has, which is structural and slow and nothing like the narrative memory that wakes up in the morning and tries to account for the night. The moment before waking will come again. Some morning not far from this one, the dream thinning at its edges, the body beginning to return to itself, the room's particular darkness starting to register again as a room rather than the backdrop of somewhere else entirely. The alarm will sound, the dream will be gone before the hand reaches to silence it. And in the prefrontal cortex, in the frontopolar tissue that spent the night partially lit, in the white matter tracks that carried signals between regions that don't usually speak so clearly, in the synaptic junctions along circuits that were activated by fabricated experience and consolidated by the same machinery that consolidates real experience, something will be fractionally different than it was the night before. Not memorably different, not noticeably different, just different, in the quiet, cumulative way that all biological change is different, below the threshold of awareness, ahead of any account the waking mind can give of it, already done by the time the eyes open and the dream is gone. The body already knows, the tissue already moved on. The exhale is longer than the inhale. It has been all night. The body, without being asked, gives more back to the air than it takes, a slight asymmetry built into the respiratory cycle, the diaphragm releasing more slowly than it contracted, the lungs emptying past the point where any effort seems necessary. You didn't arrange this. It was already running when you arrived here, and it will keep running past wherever this ends. The biology has been patient. Everything covered tonight, the prefrontal flicker, the gamma signature, the amygdala running its ancient check, the synapses adjusting to what the dream contained. All of it was happening in a body that was also just breathing, just maintaining temperature, just keeping the heart at its quiet, unremarkable pace. The cellular work and the ordinary keeping alive work running in the same tissue, on the same night, without distinction. The body doesn't separate the extraordinary from the routine, it just continues. Somewhere in the brainstem, the reticular activating system, the loose cluster of nuclei responsible for maintaining arousal, for keeping the cortex alert enough to process the world, is dialing down, not switching off, dialing. The transition is gradual and chemical, adenosine accumulated across the day, pressing gently against the circuits that keep waking going, the balance tipping slowly towards sleep's particular organization. The cortex is already receiving less. The thalamic relay, the sensory gating, the slow withdrawal of the world's insistence. It's underway. Has been underway for a while now. The sounds that were present an hour ago are still present probably, but arriving with less authority, less demand for response. The brain is already deciding at a level below decision that they don't require anything. The breath moves. The chest rises fractionally, pauses at a height that requires nothing, falls. The prefrontal cortex, which spent portions of this narration being described in some detail, its metabolic expense, its role in metacognition, its strange persistence in certain sleeping brains, is quieting. The supervisory function stepping back from its threshold, the checking and comparing running slower then slower still. Most of what was explained tonight will not be remembered by morning, not because it wasn't real, because memory is selective and sleep is long, and the brain has its own sense of what needs keeping. The feelings may stay longer than the facts, the particular quality of this hour, the weight, the breath, the darkness that has become simply the condition of being here. This may last into tomorrow in ways the specific biology won't. That's fine. The tissue retained what it needed. The breath again, longer out than in, the body giving something back. The narration is already loosening its own grip on this. The sentence is thinning, the gaps between thoughts widening, the voice that has been here all night becoming harder to locate as a separate thing from the breathing and the weight in the dark. It was always close to the body. Now it's the brainstem continues. The heart continues. The exhale, longer than the inhale, continues without you.